All right, so like I was saying, we have lots of other things going on. Like if we open up the brain here, there's all these other structures inside, and then all these hollow spaces that are there. And it's really hard to really get a sense of all these different parts and pieces just from looking at an adult brain, because all you see is this thing that looks like a big walnut. So when I introduce the brain, those of you who've been in anatomy, you've probably seen this. I like to start with the embryological development of the brain. Like, how does the brain start? And then you can see the different parts before they get all overgrown and folded in on each other. So you get a better sense of how they all connect to each other. So yeah, and I usually draw this, but I'm also just kind of worried about the times. We're like running out of time. So I'm going to see if we can speed this up using this. So 20 days. So you're only like 3 weeks post-conception, and you're already like kind of like a little frisbee of cells. You know, at this point, you've already gone through gastrulation. You've got your endoderm, mesoderm, and ectoderm, which should all sound familiar from biology. The ectoderm, which is that outermost part, is gonna, first it has a groove called the neural groove, and then it actually folds in on itself. So we get this thing called this neural tube. So right now you are a little frisbee of cells, but you have this little dorsal hollow nerve tube, this neural tube, which is going to become your brain and spinal cord. So it matters that it's hollow. This neural tube is hollow. So as it continues to develop and becomes that big, crazy looking walnut thing in your skull, it's still going to have hollow spaces. It's going to have the ventricles and the aqueducts and stuff with cerebrospinal fluid flowing around. And the spinal cord will have its canal, hollow canal with cerebrospinal fluid. So we will be following the hollow spaces as well as following the neural tissue as it gets more elaborated into the cerebrum and the cerebellum and the hypothalamus and all of that stuff. So let's continue here. So here we have, in fact, pointer options. Let's see a different color. So here we're a month in. So this is a month post-conception. That thing that started as that little tube has now got different areas that are swelling. This is obviously near the head area. So this part here, which is the forebrain, or if you've been in anatomy, the prosencephalon, the midbrain, or if you've been in anatomy, the mesencephalon, and the hindbrain, or if you're in anatomy, the rhombencephalon. Those are going to be the parts that are in your head that are going to become the brain. And then the rest of the tube is going to continue going down your spinal, the canal in your spinal column to become the spinal cord. And again, it's hollow. There you can see it says lumen of the neural tube. So this forebrain, midbrain, and hindbrain also have these hollow spaces that are going to continue on being the ventricles, like the lateral ventricles, the fourth ventricle, stuff like that. And then what we can do is talk about what do each of those different areas become. And let me change this color here. Let's maybe make it light green. So like this forebrain. This forebrain, forebrain is going to become the diencephalon and the cerebrum. So the cerebrum, this is the part that's going to fold over and become those big walnut-looking things on that cover everything else and make it kind of hard to see everything else because the cerebrum kind of overgrows. Like, you know, eventually the cerebrum is going to like be like this big mushroom cap that just folds down over and kind of obscures all the other structures. Part of the reason why I'm kind of introducing the brain this way is because we can still, this way we can see, oh yeah, there's a lot of things getting obscured as the cerebrum overgrows everything. The diencephalon, which is kind of the gateway up into the cerebrum, that is going to become like the thalamus and the hypothalamus and the epithalamus. So we will spend more time looking at the diencephalon. But again, the diencephalon is, it's kind of hard to type here, like the thalamus and the hypothalamus. So we'll be looking at that. We can change our highlighter color, but maybe make it red. The midbrain. This is going to be these different structures like the superior and inferior colliculi, substantia nigra, things that are all, they're typically involved more with reflexive stuff. So we will follow the midbrain as well, different structures in the midbrain. And again, the midbrain is below the diencephalon, which is below the cerebrum. And then the hindbrain, I can change my color here one more time. The hindbrain becomes the medulla oblongata, which is kind of this long section here, kind of that connects right before the spinal cord. It's also the pons, which is this part that kind of hangs out in front of the medulla. And then the cerebellum. The cerebellum, which is, this picture doesn't show as well, kind of hangs off the back of the brainstem. So if you look at this picture now you can see the cerebrum, the cerebral hemispheres have basically folded over almost everything. You can't really see the diencephalon. You can't see the midbrain because all of this cerebrum has folded over it. You can get a little bit of the pons and the medulla, as well as the midbrain, are called the brainstem. Brainstem is the medulla, the pons, plus the midbrain. So these things plus the midbrain. Cerebellum, again, is not officially considered part of the brainstem because it's kind of hanging off the back of the brainstem. I should also, let me point to options. Let's erase. Cerebellum and cerebrum sound pretty similar, so don't get them confused. Cerebrum is the thing that comes from the forebrain that ends up becoming this big, complicated, wrinkly structure as it grows and grows, you end up getting a surface area about the size of a tablecloth, all wrinkled up and shoved into your skull. Cerebellum is on the back of the brainstem here. And again, you can see the spinal cord, it just comes off of there. And then just kind of also making it clear, this is representing the ventricles. As all that folding is happening, that hollow space is still there and gets all kind of distorted and warped in that process. So the hollow space in that hollow neural tube that we saw embryologically, now it's got different regions. For our class, you do not need to know the names of the regions. In anatomy, you had to learn them all. The lateral ventricles, the aqueduct of Sylvius, the third ventricle. This is actually the foramen of Monroe. Here's the third ventricle, the aqueduct of Sylvius, the fourth ventricle, the central canal of the spinal cord. Now, don't worry about the names of the different ventricles and aqueducts for our class. You should just know that there are these hollow spaces that are inside the brain and spinal cord. And we're going to be looking at them in more detail in just a few moments. All right, so what we're going to do now is we're going to look at the brain. We're going to look at the different membranes that kind of surround the brain and spinal cord and kind of anchor and protect it, and talk also about the whole idea of the flow of cerebrospinal fluid. So I'm going to go back to a normal way of presenting. So again, we had this, forebrain, midbrain, hindbrain from our little dorsal hollow neural tube. And again, this part here, that forebrain, ultimately became these. What I'm doing now is a coronal section. Coronal section is like if I cut through this way and your face fell off, right? So you can see the two hemispheres of the brain in this section. This is like here, this is coronal section is cut right through here. Like your face falls off and then you see the two hemispheres, kind of the cerebral hemispheres. Hemispheres. Let me spell this properly. And then kind of going down into the diencephalon, the thalami and the hypothalamus. Going down into the midbrain. And the midbrain is just going to be this kind of midbrain area. The hindbrain is going to become the pons and the medulla. But let me make these all black because I want to use my colors, save my colors for other stuff. I should also mention the cerebral hemispheres. As the cerebrum grows and gets all shoved up into your skull, it gets really wrinkled up. in order to increase the surface area, you get little folds. So let me show you, give you some names for those, because sulcus and gyrus. Sulcus is a little valley. Plural is sulci. Gyrus is a ridge. Plural are gyri. So what you actually see is all of these sulci and gyri when you look at the right, that's all that kind of wrinkled up kind of stuff that you're seeing. All right, so there's this. This is the brain tissue that I've drawn. Then, like I said, there's the hollow spaces that are inside that we need to keep track of. Again, you don't need to know the specific names of them all. If you remember, if you were in anatomy, you learned about the lateral ventricles, the foramina of Monroe, the third ventricle, the aqueduct of Sylvius, the fourth ventricle, the central canal down the spinal cord. These are all hollow spaces and filled with cerebrospinal fluid, which we're going to describe in more detail. Let's do blue. And they're called ventricles for the big ones. We'll talk more about cerebrospinal fluid in just a few moments. Let me also just kind of improve my picture here by giving the brain tissue some. All right, so here's where we left things after the brain kind of developed. Obviously, the brain is in the skull. Then again, you have the foramen magnum, and then you have the vertebrae with the little canal going through to allow the spinal cord to go on. What kind of glial cells are going to line all of our hollow spaces?
Ependymal?
Exactly. These are all lined with ependymal cells. And I don't know if you remember it, ependymal cells actually have cilia. So that actually kind of helps kind of circulate all of the cerebrospinal fluid around in there. The ependymal cells are also going to play a critical role in actually generating the CSF. In fact, let me just describe the structures that generate CSF. So CSF, let's look a little bit more. I can use my little keyboard here to cerebrospinal fluid, and it is produced in these structures in the ventricles called choroid plexuses. So let's talk about what is a choroid plexus. So in general, if you are trying to create some fluid, the place where you've got a lot of nice isotonic fluid flowing everywhere is your bloodstream. So a typical way to create some kind of fluid is to have leaky capillaries where the fluid can leak out. So we're going to have these leaky capillaries. Which we're going to learn more about later, the idea of fenestrated capillaries to allow stuff to leak out. And then you have the ependymal cells, which are actually on these as well, which are going to adjust the composition of the leaked fluid. So it's not just going to be kind of the leaked fluid from the plasma. These little ependymal cells will help monitor and adjust the composition. But basically, you've got salty water leaking out of this structure because they're leaky capillaries. It's very similar to plasma. It's not exactly, like I said, the ependymal cells adjust it, but that's what a choroid plexus is. It's this little place where you've got fluid leaking out of the capillaries to create, this is the CSF. CSF is that fluid that's leaking out there. And we've got these in a variety of places in the brain constantly producing CSF. So if we go back to this picture here, there's choroid plexuses at the floor of the lateral ventricles here. There's a choroid plexus here in the third ventricle. There's another choroid plexus here in the fourth ventricle. So we've got these little things, these little structures constantly producing CSF, and they produce quite a bit, like I think like 100 mils every like 5 to 8 hours. So they're constantly making this CSF, and if we did not drain it, your brain would just start blowing up like a big balloon. Because it's like I said, it's being constantly produced. So what we're going to talk about now is the whole system of CSF circulation in the brain. Here we're watching it being created, but then we're going to watch it flow different places and ultimately be drained. To do that, we need to talk about the different membranes surrounding the brain. So let's do that now. So I'm going to assume you know this is the skull. So the brain, so I can erase this as well because you don't need this here anymore. So we're going to talk about now are the meninges. The meninges are the membranes that surround the brain. There is a singular, you call it a meninx, but you don't usually use it that much unless you're like in an anatomy class. So there are three main meninges. The first one, the reason I've been saving colors for myself, the first one is going to be called the pia mater. Pia mater is against the actual neural tissue of the brain. It's right on the brain and it follows every little curve. If there is a gyrus, it'll go up. If there's a sulcus, it'll go down. It's this kind of connective tissue that is just right on top of the surface of the brain tissue itself. It would be like, I don't know, like wearing some really tight Lycra workout thing where it's like, yeah, you've got clothes on, but you look the same as if you didn't wear clothes except for having color on you or something, right? It's really just right, hugging every little curve. The pia mater. The next one is called the arachnoid layer. And it's not actually called mater. Pia mater means faithful mother, because it's like faithfully following every little thing, every little curve. Arachnoid layer, you're going to see, gets the name kind of arachnoid from spider, because it has these little kind of web-like struts that anchor it to the pia mater, actually. So the pia mater is anchored, and if you take a look, it does not follow every little curve and nook and cranny. It actually, let me try to just draw this and then I'll continue talking. So this arachnoid layer, maybe I'll also use my, what I'm drawing now is called the subarachnoid space. It's that space underneath the arachnoid layer that is being kind of reinforced with the little struts that I drew. So that is the subarachnoid space. The subarachnoid space is going to be a place where the CSF actually gets to drain from. So if we go into the, we look here, this, again, this area, hold on a sec. Oh, here, this. Oh, here, I've got my little pointer thing right around here. This is the fourth ventricle. There are going to be these little connectors. The lateral apertures of Luschka and the median aperture of Magendie that connect the ventricle to the subarachnoid space. So now that cerebrospinal fluid that's inside can get outside and get into the subarachnoid space. So this, maybe I can use my typing tool because it'll be easier. Actually, this is smaller. Connects to the fourth ventricle. The median and lateral apertures. You don't need to know the names of the median and lateral apertures, or again, like I said, when I took this class, it was the foramen of Magendie and Luschka. Median and lateral apertures to allow CSF flow.
Can it flow both ways, or is it just out of the ventricle?
It's because of the constant production of the CSF by the choroid plexuses. There's a positive pressure where it's always going to be leaving the ventricles, right? Because it's basically, there's not necessarily a valve there, but you just have a constant buildup of pressure inside. So you've got this little hole so it can get out. So now the subarachnoid space is underneath or inside the brain, but also outside the brain. Basically, what this gives us is kind of almost weightless brain. If you think about lying in the bathtub, especially if you've got some bath salts in there, part of why it's so relaxing is like you don't even realize you have a body. You're just kind of floating weightless in there. Like if you've ever like taken a bath and just, I do this sometimes just for fun. It's like you kick out the stopper and you let the water drain, and all of a sudden you touch, you feel the bottom of the tub, and then you really feel the bottom of the tub. And then it's like, holy crap, I'm like 150 pounds lying on the bottom of the tub here. Right. But when you're like floating in water, you weigh, just like a few percent of that overall weight. So the brain is delicate and squishy. If you get like an actual brain, that people talk about it being the consistency of like soft tofu. It's really delicate. And it would just be squishing itself under its own weight. A brain weighs like a few pounds, but because it's basically inside and outside, completely enveloped in salt water, it's basically floating around and only weighs a few percent of its actual weight. So that it helps kind of make it weightless and kind of cushions it inside your skull there. Obviously, we're not quite done with our story here because we need a place for it to drain from the subarachnoid space, or else the subarachnoid space is ultimately going to inflate and pop. So we need to add the last of the meninges, which is going to be the Dura Mater, or the hard mother. So again, we had the Pia Mater, the arachnoid layer with its subarachnoid space. Now we're doing the Dura Mater. These are the three meninges. The Dura Mater is very leathery. If you've taken anatomy again, you've seen it's kind of like almost a swimming cap that's over the brain. It's tough. That's why it gets its name. Dura Mater means hard mother. And at least in the skull, it has these little, what are called dural sinuses. These little, now I wish I could bring this down. Is it going to be possible? Hold on. Oh, that didn't work. Hold on, I'm trying to do something here. Oh, this isn't working. All right, so Dura Mater has what are called dural sinuses. These are like little canals. Within the dura. The way they come about actually is in the skull, there are two layers. There's a periosteal and a meningeal layer of the Dura Mater. So we have one layer, we have the other layer. This is my dura. Most of the time, you don't even know it's two layers because they are fused and it just is like one layer. But there are places where you get that little opening, and that is my dural sinus. And there's a bunch of them. There's a superior, again, in anatomy, you had to learn about the superior sagittal sinus, the inferior sagittal sinus, the transverse sinus, the straight sinus, the occipital sinus, all these sinuses. They're basically little tunnels in the dura. And then this is on top of the arachnoid layer. So like if here's my brain, here's the arachnoid. This is the subarachnoid space in here. Which maybe I'll color it in a little bit with some green. Right. So is everybody basically, I've taken this little spot right up here where we had the dural sinus, the Dura Mater, and we got the brain, and we've got I've taken this and I've kind of blown it up so you can see it in more detail. And what's going to happen is there are going to be these little things called arachnoid villi. In fact, let me use my typing, so this is going to be better. So basically, these arachnoid villi are how we're going to drain the CSF that's in the arachnoid space, subarachnoid space, into the dural sinuses. And then these dural sinuses, these all ultimately drain into the jugular vein. So the jugular vein, it's the main vein that's draining your head. So all of these dural sinuses, all of these tunnels in the Dura Mater, all ultimately converge. There's a confluence of sinuses, and they all ultimately just dump into the jugular vein. So now if we go back to our other picture here, we can take a look at what's happening here. We start here, choroid plexuses, we're making CSF. And then that CSF is flowing around in these hollow spaces, circulated around by the little cilia on the ependymal cells, but ultimately we have these little apertures that then have that CSF go into the subarachnoid space. So that would be like 2 going into the subarachnoid space. But then from the subarachnoid space, we have those little dural sinuses that we can get to through the arachnoid villi. So 3 is going to be going into the dural sinuses via arachnoid villi. And then 4, what's going to be the final destination for the CSF?
The jugular vein?
Exactly, the jugular vein. Right, and like I said, CSF is basically like plasma. So dumping some CSF and just adding it into your blood, it just like spit in the ocean. It just like disappears. Just adds back into the original, fluid that leaked out at the choroid plexus to start with. So you should be familiar with the basic flow of CSF. Being produced in the choroid plexus, going from the choroid plexus into the subarachnoid space via the little apertures, then through the arachnoid villi, getting into the dural sinuses, and ultimately from the dural sinuses, draining into the jugular vein. And again, this is happening constantly. Like I said, you've got like 100 mils or so every 5-8 hours getting produced. What happens if this gets blocked? The drainage, the flow gets blocked.
Your brain explodes.
Well, your brain gets under pressure minimally. Hydrocephaly. It's like let me go. Oh, actually, I want. So especially if it happens when before your skull bones have fused and the things are still soft, right? Babies' skulls have the fontanelles, are kind of squishy. You get, this kind of thing. You get where the skull just starts kind of inflating. Oh, here, right? So if this is happening, they've got to go in, they put in little shunts to restore the proper drainage of the CSF. So, it's obviously important that right, because normally you have the ventricles, you have the ventricles, but if the ventricles aren't draining, they're going to start inflating in there. So again, it is kind of like the brain is kind of blown up like a balloon like that. So, and they can treat this kind of stuff but there are times when they have to surgically and put in shunts to keep the drainage going. The last thing I'm going to do before we take our break is just give you the concept of the spinal lumbar puncture. Let me do that, and then we'll take a break. So the basic idea of the lumbar puncture is all of those meninges that we were talking about and all of the flow of the CSF is not just, it's not just the brain, it's also the spinal cord. So you have that going here, but then you have the Dura Mater goes all the way down to the very bottom of the spinal column. And it's filled with CSF. So if you want a sample of somebody's CSF, what you would like is to get some CSF, but you'd rather not drill a hole in their skull to get the CSF out of their subarachnoid space or their ventricles. So it turns out that you have the CSF continues all the way to the bottom, and it has this area where it's called the cauda equina, where you just have a bunch of nerves rather than the spinal cord. The spinal cord is, you don't want to stick a needle in here because you might nick the spinal cord and cause damage. But you could take a needle below lumbar 2 or so. You can wiggle between the vertebrae through the disc, intervertebral, actually through what was called, what do they call it? The epidural fascia. And you get through the Dura Mater, get in there, and this is actually CSF. You can suck a little CSF out, you know, in a relatively non-invasive way. You don't have to go into their skull or anything. So you can suck a little CSF out. You can analyze it, see if there's any blood or pathogens or whatever in there. You know, and then they have to kind of lie down and hopefully the little hole seals up and the CSF regenerates. Supposedly, if you stand up too soon after one of these procedures, you get just excruciating headaches. But because the Dura Mater extends all the way down to the bottom of the spinal, not just the spinal cord, but the spinal column, and that subarachnoid space is filled with CSF all the way down to the bottom. And because you just have these little wiggly nerves down here that are, you know, pretty safe to stick a needle in the vicinity of without hurting anything, this is a nice way to suck out a little CSF and analyze it for a, you know, kind of clinical, if it's clinically prudent. So lumbar puncture, what's the other word for a lumbar puncture?
Spinal tap?
Spinal tap.
Spinal tap, yeah, spinal tap. So spinal tap or lumbar puncture, the same deal. All right, so we have talked a bunch about the brain as kind of more kind of the support and sustenance of the brain in your skull. Now, when we return to the brain, we'll be talking more about kind of what it's doing and how it processes information and things like that. Are there any questions up to this point?
I just had a question. So the arachnoid villi is when it drains from the subarachnoid space into the dural sinuses?
Correct.
Okay, thank you.
Yeah, so they're like these little valve-like structures that go drain from subarachnoid space into the dural sinuses, the little tunnels in the Dura Mater, and then all of those dural sinuses ultimately dump that fluid into the jugular. So that's the end of the flow. Starts at the choroid plexus, ends in the jugular vein, and just keeps going, you know, kind of repeat until dead.
Thank you.
Sure. All right, so let's take a break. It's 9:43. Let's come back at 10:15,